Today I am going to explain about tissue culture techniques, types and its application. There are different types of plant tissue culture. They are like anther culture or ovary culture, protoplast culture, seed culture, embryo culture, stem culture, bud, callus, cell suspension culture. Process of plant tissue culture. First, we have to select the parent plant. From the parent plant, selection of the explant from mother plant will be done. Later on, it will be inoculated into the medium, usually Murashige and Skoog medium. Later on, this culture will be incubated in optimum temperature and light for shoot initiation. Later the callus induction takes place, then shoot formation takes place. These shoots are transferred to rooting medium, which will be having a higher concentration of auxin. Later the plants are kept for hardening in greenhouse, sold to nursery and then potted up. Protoplast fusion. Now, what are the applications of protoplast fusion? It can be used for the production of somatic hybrids or cytoplasmic male sterility line production, or even autotetraploid or allotetraploid production, heterozygous line production. Even some somatic hybrids with specific aliens can be regenerated. Production of unique nuclear cytoplasmic combination can be seen through protoplast fusion. Next is somoclonal variation. It plays an important role in polyploidization. Genetic variation can be raised, which include resistance to particular disease like herbicide resistance or stress tolerance, etc. Now in this picture you can see that two tobacco plant protoplasm obtained by digesting away the cell wall are fused to produce cells that acquire some of the characteristics of both the genetic background and can be regenerated into plant with some trait from both parental plants. Some of the application of tissue culture are micropropagation, germplasm conservation, somoclonal variation, haploid and diploid production, in vitro hybridization, protoplast fusion, embryo rescue and synthetic seed production. The rooted shoots are potted up, which is called as deflast and hardened off. This is necessary because as many young tissue culture plant do not have waxy cuticle to prevent water loss. Now these tissue culture plants sold to nursery and then potted up. Micropropagation. What is micropropagation? Large-scale production of genetically identical clone in vitro is called micropropagation. Now, what are the advantages of micropropagation? Millions of shoot tips from a small piece of plant tissue in a short period of time and space will be produced. Next is maintenance of genetic uniformity in the progeny can be done. It can produce exact copies of plant required that have desirable traits. Even supplies plant throughout the year irrespective of seasonal variation. These are the advantages of micropropagation. Next is a commercial application of micropropagation. Micropropagation of orchid demonstrate profit all over the world. Many other crops like banana, papaya, potato, anthurium, apple, cherry, etc. have been grown through micropropagation are commercially exploited. Even development of disease resistant crop can be done. Some valuable clones recovered from virus such as tobacco mosaic virus, sugarcane mosaic virus are also be done. Among innovative techniques of micropropagation, the concept of somatic embryogenesis with synthetic seeds, production of artificial seed technology are very promising. Germplasm conservation. Maintenance of germplasm of plant vulnerable to pathogen, insect and various climatic hazard and plant with early embryo degeneration or difficult to maintain. Plant species which are endangered, rare, threatened with extinction are needed to conserved by ex situ method of germplasm conservation. Provide cost effective alternative to growing plant under field condition, nurseries or greenhouses. And cryopreservation of germplasm is really effective in conservation biotechnology. Next is embryo culture. Using embryo culture, seed dormancy can be overcome. Embryo rescue, which can circumvent post-zygotic barriers to raise rare hybrid by rescuing embryo from incompatible cross is most popular application. The production of embryo from somatic or non-germ cells usually involve callus intermediate stage, which can result in variation among seedling. Artificial seed production. Synthetic or artificial seed, living seed like structure, they are called as like living seed like structure, or any totipotent cell, like for example somatic embryo or meristematic tip, etc., which is artificially encapsulated by chemicals like hydrogel, which behave like true seed if grown in soil and can be used as substitute of true seed and possess ability to convert plant ex vitro or in vitro. Now, what are the uses of haploid or diploid production? Here, these are produced by anther or ovary cultures. The production of pure homozygous line in less time period will be done through this culture. Androgenic haploids are used to produce different lines of aneuploidies like monosomic, nullisomic, trisomic, etc. Induction of mutagenesis can be done. Even recessive trait like low glycosinolate in brassica can be made expressed. Use of hybrid development and early release of variety can be done through haploid or diploid production. First, the plant of our interest. From plant of our interest, we have to isolate the anther that will be cultured in a medium. It might produce either a callus or an embryoid, but both will finally give rise to a haploid plantlet.